And now we're going to go into our first session, which is then imaging across the scales. And so we're going to start off at the very small scale with single molecules, and Andrew Carter from the LMB is then going to tell us all about Shulin, the novel factor for axonemal outer dynein arms. So if you could share your screen, Andrew, then.
I was just having a quick look on the web. I guess everyone knows, but the Nobel Prize went to Charpentier and Doudna for CRISPR, which is very exciting. So try and overcome that excitement. Right, so what I'd like to do today is to tell you about a story that I think is one of the broadest that's come out of my lab. It spans all the way from identifying a novel protein, finding out what it does in delivering things to the cilia, to cryo-EM structures that find out how it works. And for today's meeting, I think perhaps the most relevant thing will be that it's some nice examples of how to use cryo-EM for looking at really quite flexible samples. So this work is carried out by 3 postdocs in my lab. It was really started and spearheaded by Girish Mali, and then Ferdos, Abid Ali, and Clinton Lau have done the cryo-EM, the higher resolution cryo-EM work. So I should start off by introducing you to cilia. There are 2 types of cilia: primary cilia found on all cells, usually just a single version of it, and then motile cilia, which are often found in patches. For example, in this image below, you can see this carpet of motile cilia. They're based around a microtubule structure called the axoneme that's embedded in the centriole that makes up the basal body. Their key function, of course, is to move. And what this video shows is some ciliated epithelia, and you can perhaps make out that there's this flow of material past these continuously beating cilia. So cilia are found throughout the organism. They are found in the brain, where they circulate the CSF fluid. They're found in developing organisms where they help set up our left-right asymmetry. So patients with defects in cilia can actually have the left-right asymmetry reversed. They're very important in the fallopian tube for moving implanted embryos around. They're involved with the swimming of sperm. And then perhaps the most important function day to day is clearing things from our lungs. So they're involved throughout our respiratory tract and are able to shift things like bacteria, etc., out of there and keep those clear. And problems in motile cilia and mutations in the components cause a number of different ciliopathies, of which PCD is one of the most common, which sort of illustrates their importance. Okay, so the movement that I just showed is driven by motor proteins of the dynein family. And if you take a slice through the cilia, you'll see that these dyneins are arranged. There are a load of dyneins arranged on the outer ring. These are the outer arm dyneins. Looking here at the family of heavy chains, the outer arm dyneins consist of either heterodimers or heterotrimers, depending on the species of these central heavy chains. Then on the inner ring, there are these inner arm dyneins, and there are lots of those, and they're established in a large array that's repeated throughout the length of the axoneme. So far for the dynein family, we have structural information from cryo-EM of the cytoplasmic dynein 1 and 2. These are involved with transporting cargoes. But so far, no structures of the axonemal dyneins, although I'll tell you about the one today. So today's talk will focus on the outer dynein arms, also called ODAs. We're going to talk about this 3 heavy chain, 3-headed motor. And you can see it's a complex both of these heavy chains, but also of intermediate chains and a whole set of different light chains. So one of the things that Girish did when he came to the lab was spark our interest in the whole assembly process of dyneins and how they get to the cilia. So it turns out that after synthesis in the cytoplasm of the different outer arm dynein components, there's a whole range of different chaperones and factors, some 10 different proteins required to assemble this complex. And then these outer dynein arms can hook onto the intraflagellar transport machinery that's driven by kinesins on the way out and a dynein 2 on the way back. And that can deliver them to the cilia so that they end up in their end array. But the missing question that I think has interested the field for a number of years is there anything else happening in between to these sort of two assemblies and delivery? Are there any other factors involved? And so what we wanted to do was to set out to see if we could find out whether there was any involved by trying to find out what was bound to the dyneins after they've been synthesized, but when they're still in the cytoplasm, before they've got delivered to the cilia. So in order to do this, we turned to a ciliate protozoa called Tetrahymena. Now these use their cilia to swim around, and they're pond-living organisms. And the great benefit of them is you can grow them in a flask, so you can grow litres and litres and get a good amount for biochemical material. It's also possible to genetically engineer them. You do this using a bialistic transformation, so you mix DNA with gold beads and fire it into these Tetrahymena. But they're interesting organisms because their nucleus, their macronucleus, they actually have two nuclei, complete sets of genomes, one in the micronucleus, which is mostly shut off except for during mating, and then the macronucleus, which sort of drives everyday functions. And in the macronucleus, you have 45 copies of every chromosome, and there are hundreds of chromosomes. All the DNA is fragmented up into these sections. So in order to do homologous recombination, you introduce your DNA, but then you have to select for quite a time to slowly replace all the copies from 1 up to sort of as many as you can get, but towards 45 copies replaced. So the way that we approached this project was to make a tagged intermediate chain. And when you look at Tetrahymena with a flag tag antibody, you can see that it all goes into the cilia. Now, because we wanted to look in the cell body, we deciliated the organisms using dibucane, which is an anesthetic, and the cilia drop off. And then if you wait some time, you can see that the cilia start to regrow, a massive amount of ODAs are produced, and they start accumulating in the cell body. We then used gel filtration to purify out the larger complexes that we knew corresponded to the assembled dyneins and did mass spec to identify them. And when you look at the trace that came off gel filtration, it's a single peak, which is good. And we found two factors that were bound in pretty stoichiometric amounts to the ODA motors. So this top one was called MS1. It's a very Tetrahymena specific protein, although it contains a conserved domain. And then YU3, this lower one, actually has a very good human homologue, this C20ORF194 protein. So that looks like it's conserved across species. So the next thing then was to try and ask whether these proteins not only bind the ODA, but are also important for getting them to the cilia. And so we made knockouts by using the same sort of bialistic transformation. And when we did that, we found that the swimming speed of these Tetrahymena is severely decreased for both mutants, suggesting that there's a defect in the cilia. And then we stained the Tetrahymena for the outer arm dyneins. And what you can see in this panel is that in the wild type, the cilia are all full of outer arm dyneins, whereas in both of these mutants, there's a real lack of it. So showing that by getting rid of this protein, either of these factors, you reduce the amount of ODAs that can get into the cilia. Okay, so we wanted to try and look next at what these proteins are actually doing to the ODAs themselves. And in order to do that, we wanted to sort of take a system where we could reconstitute dyneins with either one or both or either factor. And so to do that, we purified our dyneins from the cilia themselves. You can do that by dropping them off with dibucane and then purifying out the ODA protein that lacks any of these factors, and then adding them back. And what this gel filtration trace showed was that in the presence of these factors, you can get them bound to the complex. So here you can see the gel from the peak fraction, and you can see both proteins bind. And either protein binds on its own, they don't require each other to do that. So that allowed us to look and see what they were doing. And the first thing we assayed was how well the ODA, the dynein, was able to move microtubules in a microscopy-based gliding assay, where the microtubules move over the surface of the cover slip, driven by dyneins. And the result from that was that the YU3 protein, at least, is a very strong inhibitor of this ODA. It seems to shut off its activity. The MS1 was not quite so clear, possibly less of an inhibitor, and maybe working in a different way. So what this suggests then is that the function of at least of this YU3 protein is to inhibit the dynein, and that somehow is involved with stopping it getting to the cilia. Quite how that works was not clear yet. It may be that it stops it binding inappropriately to microtubules in the cell body, or perhaps it stops the dyneins when they're being dragged into the cilia, sort of running straight back out again. But what we wanted to do next was to ask how they were having this inhibitory effect. And so what we did is took the complexes that were made and looked at them by negative stain electron microscopy, where you can collect lots and lots of images, average them together, and start sorting particles into different conformations. And when we did that, with the ODA, the dynein on its own, you can see that you always get 3 different motor domains visible in these averages, these small classes. So they're in this very open conformation. This is ODA that's been purified from the axoneme. But when you start adding the factors in, you see a much more frequently a closed conformation, where you only perhaps see 2 of these motor domains, and they are about clustered together in a cluster, as shown in this cartoon. And what was clear from our analysis is that YU3 is actually able to do the clustering on its own, and is actually stronger on its own than when you have both proteins present. So we then took the liberty of renaming this YU3 protein Shulin, and that comes from the Sanskrit word meaning the one who holds the trident, i.e. keeping the 3-headed motor together. So what we wanted to do now is try and use cryo-EM to see if we could get a sort of higher resolution view of how this protein is working to cluster these motor domains at a molecular level. And just to take you through the rough strategy, there's a lot of images on this slide, but we have these images of the ODA Shulin complex in ice. We pick them, we get class averages, we select good particles. This is all very standard, as you know, ways of processing cryo-EM data. And we do a global 3D refinement to get a first structure. And the resulting structure is really pretty weak, pretty poor resolution, about 15 angstroms. And that is because it's such a flexible motor. But then, as many do, you go in and mask out different subregions, and you can improve the resolution of the different parts of the structure. And what I just want to do is give you a few examples of how, by tweaking with these masks and really playing with the optimization, you can actually really improve the sort of structural information of different parts of the structure you can get out. So the first example is sort of the central region of the motor. After we classified to get the good particles and did a refinement, we then just masked out this central region, which you can see here in green, and then went through a round of different refinements that just then focus on improving the alignment of the particles to this central region. And by really playing with the mask, you can improve this process. We ended up going through about 5 or 6 rounds, each time just making the mask a little smaller to get rid of any flexible bits, or if there looked like there was a region that was a bit more ordered, then we'd include that again. And playing with the softness of the mask and really sort of just playing with it and getting it to go. And by this, we were able to then push the resolution to 4.3. And in the core, of course, it was really very good. It was good enough to be able to build de novo chains, and I'll tell you a bit about that in a minute. The other example is the use of signal subtraction, also commonly used. So in this case, we had a part of the structure which from the sort of first global refinement was very weak and was not showing up. And part of the problem here is that the rest of the structure, which is well ordered, tends to dominate it and prevent alignment on that flexible region. So by subtracting out that, by making a mask and then subtracting everything else, we were able to sort of improve that resolution. And then by sort of doing sub-masks and again, playing with them and optimizing it, we were able to then push these different regions that have been very flexible to high resolution. And the trick here was that actually you couldn't just go from the first mask, you couldn't try masking out in this case in two separate bits. You really had to do this in a two-step process. Okay, so the result of this work was a map that was made up of lots of different parts where we have a structural information where we can build and assign chains in some parts, and in other parts we're really having to rely on homology models based on other known Dynein structures. But the resulting structure is shown here. This is the Dynein outer Dynein arm. You can see the three different heavy chains, which we call DYH3, 4, and 5, here in pink, dark pink, and blue. And if I play the movie, you can see the Shulin in green down here, all of the intermediate chains in yellow and red, and then the different light chain towers up here. So let's try splitting that apart a bit then to sort of look in a bit more detail at this structure. In the core of the structure, there is this DYH3, DYH4 chains. These are the ones that are conserved across species. And their organization is actually pretty similar to the cytoplasmic dynein that I mentioned before. They're these long chains, they've got these long tails that are held together with this dimerization domain at the bottom. And then there are intermediate chains that bind here. And the differences are that this outer Dynein arm lacks the light intermediate chains. Instead, it has a light chain on just one of the two chains. And then it has a much more complicated tower of light chains. So instead of just the three dimers found in cytoplasmic dynein, you've really got this very elaborate structure. Just to zoom in on that, you can see here the different subunits that we were able to identify, and each of these different subunits is a different light chain. There are a whole set of heterodimers, and because of the way they're arranged, this final bit sort of bends over at the top. So the other really interesting thing about this is the third heavy chain, the DYH5. And for a long time, I'd wondered how that was going to be connected to the rest of the molecule. And it turns out that actually it's much shorter in some ways. It's the helical bundles that make up these long parts of the chain. It's about half the length. And they're replaced at the end, instead of this dimerization domain, by this Kelch domain. And it just sort of docks on to one of these other heavy chains. And so this is really a very beautiful evolutionary case where you need the extra motor. And so you sort of just plonk it on and dock it onto the other structure. Okay, let's look next at the Shulin molecule itself in the center. How is it acting to inhibit this structure? So when you look at Shulin, it's also another evolutionarily interesting molecule, because the first half of it is related to a histone chaperone called the FACT complex, a eukaryote. And the second half is actually much more similar to a bacterial GTPase. And then at the end, there's an extra domain that's added on, this so-called finger domain that sticks out. So the way this protein works then is to actually stabilize the ODA in a conformation where all of its motor domains, which are these top bits here, are clustered together. And they're clustered together by forcing them into a conformation that we call the pre-powerstroke. That's one of the states of the catalytic cycle. So in dynein, you have the stalk, which binds to with the microtubule binding domain at the end. You've got the linker, which bends and straightens during each step of the motor walking along the microtubule. And then you've got an ATP binding AAA ring in the middle. And you can perhaps make out that all of these linkers are held in this bent, 90-degree bent conformation. And all that does is allow these motor domains to stick together, and all of the stalks are pointing out in the same correct direction. Now, the stabilization occurs because the Shulin binds to all of the different chains. It makes contacts to DYH5, DYH4, although these are pretty minimal. And then it makes really extensive contacts with the DYH3 chain in the center. And so part of its role here is just to hold all of these chains together, which then stabilize other contacts up in between the motor domains. But its other role then is to actually hold together, hold the DYH3 itself into this pre-powerstroke conformation. And it does that by binding extensively to the tail region here, and then sticking up that C3 finger to hold the head, the motor domain here, exactly in the right position. So I think this is rather lovely because it explains how this very strange factor can specifically bind and inhibit this protein. And so to wrap up, I've told you about Shulin binding, the fact that it's involved with getting the ODAs to the cilia, and I've shown a molecular mechanism for that. The question really is this protein acting during the delivery of the dyneins to the cilia? And we've recently got another tiny little bit of data that mentions this. So when we stain cilia with a Shulin antibody, we can see that during the time where there's active transport into the cilia, i.e. when they're regenerating, you get lots of Shulin there. And yet when in mature cilia, where there isn't the transport, it's missing. So that suggests it's a factor that's actually binding and going with the ODAs as they go to the cilia to prevent them perhaps running out inappropriately. And with that, I'd like to wrap up and thank the people involved, particularly Girish, who started the project, Ferdos and Clinton, who did the cryo-EM extensive studies that I talked about, the rest of my lab, mass spec facility, electron microscopy and scientific computing, and all the others who have helped the project in the way. So thank you very much for your attention.
That's great. Thanks very much, Andrew. That's brilliant. I think we've got some time now for some questions. And Chris, have you've been looking at the questions?
Yes. Can you hear me?
Yeah.
Okay, Andrew. So we have a question from Jose asking, how do you assess the influence the electrons in the EM experiment have on the structure that you observe in the end? And I think this is a general question about assessing for radiation damage during the experiment.
So I guess the answer is we don't assess directly, but we do use data collection conditions where we know that damage is somewhat minimal. And so there's generally fairly set criteria for how much dose you can give a sample before you start seeing damage. And that's been generally worked out by others. So we're not sort of specifically.
Okay, we also have another question here. Is Shulin widespreadly found in eukaryotes, or is it exclusively found in the Tetrahymena that you studied?
Yes, there's this close homologue in humans, and we haven't done an extensive study across, but yes, it is found throughout ciliated eukaryotes as well. So that's one of the things I think that's exciting about it, is that it's not just a Tetrahymena specific factor. It seems like it's going to be needed across different species. It's also there's a homologue in Chlamydomonas, which is another model system used extensively to find cilia, and people have started looking there as well, and it's present and seems to be having a similar function.
Okay, I had a question. A rather naive question, I suppose, is you saw that the interaction with the Shulin inactivated the motor component, but do you think there could also be a role in packaging the molecule for transporting it around the cell? Could that be an additional mechanism?
Yes, I think there could, and the evidence for that is that the human homologue is known to bind to a cilia-specific small GTPase called AL3. And so what that suggests is a mechanism that we haven't really been able to look into yet, but we plan to, whereby you could imagine that it's sort of targeted by its ability to bind to cilia-specific small g proteins.
Thank you.